So I've introduced T2 star changes. I have introduced T2 star. Now the question is, how do these changes in T2 star relate to something useful, to some biophysical mechanism? Now we'll start here with magnetic susceptibility, and we have we know from magnetism that the magnetic field in an object depends on the object's properties. So we have the magnetic field, and this the magnetic field inside the object is linked to the externally applied magnetic field B0 through the susceptibility. You can see this is multiplied by 10 to the minus 6, so the susceptibility here is given in ppm. So it is a relatively small effect. And the major effects that we're looking at is diamagnetism, it's a repelling force, and paramagnetism, which is an attracting force. It has to do with interactions between the atoms and the external magnetic field. So here I want to illustrate an example, and this is a levitating strawberry. It's placed on a strong magnet, permanent magnet, that is very inhomogeneous, and the strawberry is primarily water, so it's diamagnetic, and so it levitates in air here, as you can see on this video from YouTube. Here's another example, and that is a Canadian frog that is also placed on top of a strong magnetic magnet. The frog is very light, so it has the same repelling force as the strawberry and about a comparable weight. So this animal is floating on top of the magnet. We could, of course, ask the question at one point, are we able to levitate humans? But this is more of an academic interest than of reality, what we want to do. So these are the effects on two living or examples from life sciences. In the following experiments that I will be showing you the effect, the levitation effect. Then we'll look at the effect on fluids in a magnet as it's turned on. And finally, what we're going to be seeing is the difference between molecular oxygen and nitrogen, that one molecular oxygen is paramagnetic and nitrogen is diamagnetic, just like water. And we'll see these three experiments just now. So in this experiment, what we have here is a strong magnet. On top of it, a diamagnetic substance, which is held down by another magnet. So I relieve it now, and now you can see the repelling force of diamagnetism of the magnet, of the magnetism on the object. It is hovering in midair. So if I take here a piece of paper, I can easily slide it underneath. And you can clearly see how it is hovering on top of the magnet. There's no physical connection. There's a distance. That's the repelling force of diamagnetism. Okay, so in this experiment we have again an electromagnet with the poles. Between the two poles where the magnetic field is established, we have two cuvettes. In those two cuvettes, on your right side, we have water, and on the left side we have iron perchloride, which is paramagnetic. And currently the magnet is off, so you can see the level of the water, which is on the same level. And when I turn it on, the magnetic field, the paramagnetic substance is pulled upwards, and much more so than the water is pushed downwards because it's being repelled by the magnetic field. Okay, so what we have here is a magnet which is on. So it's a magnetic field created between these two poles. And we'll first pour liquid nitrogen in this experiment on top of here. And what you will see is that the liquid nitrogen will just fall through. So I'll take it now, and you can see the liquid nitrogen just falls through. As we have seen in the course, liquid nitrogen is diamagnetic, so there's no force on it. Now, if on the other hand I take liquid oxygen, I'll repeat the same experiment. And now you can see how it is being retained between the two poles by the magnetic field. That's the force of the magnetic field on the paramagnetic molecular oxygen. Okay, and a similar situation arises in tissue that we have seen now with the demonstration with paramagnetic oxygen and diamagnetic nitrogen. We have in our tissue, in our blood actually, we have two types of hemoglobin. Deoxyhemoglobin, which is paramagnetic, and oxyhemoglobin, which is diamagnetic.
This has to do with the fact whether there's oxygen bound to the heme ion or not. This changes the configuration and of the electrons, and therefore changes whether they are para or diamagnetic. So now what happens if we are looking at a blood vessel that's put into an external field B0 like this? And here we have the situation with a lot of deoxygenated red blood cells. So we have a number of red blood cells here that are deoxygenated. They are in blue. In red are the oxygenated red blood cells. The paramagnetism, the effect, the susceptibility of deoxyhemoglobin is 1.6. So what now happens is there is a distortion in magnetic field that is generated. We do know that this actually happens based on the orientation. If you take an infinitely long cylinder, for those of you who are familiar with electrodynamics or remember the calculations, if we put a cylinder, infinitely long cylinder, in a magnetic field parallel to the magnetic field, we would not see a distortion of the magnetic field like this is seen here. This one here is, however, perpendicular, and here the effects are quite substantial. There's a distortion around the vessel of the magnetic field. If we take now a oxygenated vessel, that's an artery, here we have a vein, we have much more oxygenated red blood cells and very few deoxygenated red blood cells. So the average susceptibility in the blood vessel is around minus 0.3. And here we have very little distortions of the magnetic field outside of the vessel. So inside the vessel, we have a big susceptibility effect. And here I'll take first the deoxygenated capillary. The magnetic field is along Z, so it's to the right. And I'll plot here the susceptibility as a function of position. It's a rough approximation, it's a sketch. We know that we have deoxygenated capillary, deoxygenated blood, so the susceptibility, the magnetic field inside is higher. We have the tissue, which is essentially just water, the susceptibility is lower. So we have a different susceptibility between inside the vessel and outside. What this means in terms of magnetic field is that we have a different magnetic field inside the vessel and outside the vessel, and somewhere there is a transition between the two magnetic fields. This transition is not immediate, we don't have a discontinuous nature, so it falls off gradually with a appreciable gradient. So what this means is that actually outside of the capillary we have a change in magnetic field, a change in delta B0 that's happening inside the tissue. If we look at the oxygenated capillary, I'll draw it in red here, the difference in magnetic field, that change that happens in the tissue, is much smaller because we have almost a nearly perfect diamagnetic situation in the blood here. Now the consequence is if we have such a gradient of magnetic field surrounding the capillary, the image voxel is much bigger, so this means that we are going to have a delta B that is induced by physiology and that is non-zero. So there is a consequence here that the T2 star increases with decreased deoxyhemoglobin concentration in the tissue. So that is in the blood, but that's the average deoxyhemoglobin concentration changes. If it decreases, then T2 star will increase because we are reducing this delta B0 term here, which we have seen previously influences the T2 star. Of course, a little caveat, this depends on the venous architecture in the voxel, it depends on blood volume, it depends on orientation of the blood vessels, but we'll assume here for the rest that this is fairly randomly ordered. So now, how do we get from a change in delta B0 that's induced by susceptibility changes in the vessel to a contrast that is dependent on the blood oxygen level? So we know from brain physiology, we've discussed this in course 7 of the first part of this course, that the oxygen consumption increases much less than blood flow during a mental task. The mental task can be finger tapping, it can be really a mental task, it can be looking at an image, it can be listening to a sound, etc. So what is the consequence if oxygen consumption increases less or doesn't increase, but blood flow increases? And this is also something we've discussed in course 7. is from mass balance, from fixed principle, the concentration of oxygen flowing in, the arterial oxygen concentration minus the venous oxygen concentration is equal to the rate of oxygen consumption divided by flow. So if we take the simplified assumption that situation, just for mentally thinking it, that the oxygen consumption does not change, but blood flow increases, then this difference has to decrease, and the only thing that we can change is the oxygen consumption in the veins, and this one will increase. The oxygen consumption in the veins is linked to the partial pressure of oxygen in the blood, and this in turn is linked to the percent saturation of the hemoglobin molecules. This is the saturation curve of hemoglobin here. It's a monotonous curve. And so the oxygen concentration in the blood will change the saturation of the deoxyhemoglobin in the blood. Here we have typically around 90-95%. Most of the hemoglobin is saturated, but this will change in the blood. So for now, looking at the hemodynamic response, what one sees in physiology is that there's an increase in blood flow. So there's a decrease in deoxyhemoglobin in the tissue. There's an increase in BOLD. There's, however, a counteracting effect that is also typically an increase in blood volume. This increases the number of red blood cells in the tissue. So the deoxyhemoglobin increases, and that decreases the BOLD effect. And I won't go into much of the details here. It's not the purpose of the course to be a course on fMRI, but in general, the BOLD response, the BOLD signal, is a function of, is positively correlated with changes in blood flow, inversely correlated with changes in blood volume, and inversely correlated with changes in oxygen consumption rate. This is cerebral metabolic rate of oxygen, cerebral blood volume, and cerebral blood flow. So if we look at the vessel architecture of the brain, this is what we see in an MRI. In red are the blood vessels typically. And now if we zoom into an imaging voxel, that's our MRI voxel here, then we see inside the voxel the fine architecture. So we'll see here the arterial input, and we see the venules and the capillary bed. So this is essentially what contributes to the changes in delta B0. We're looking at a voxel here. Inside the voxel we have this architecture, and this changes now depending on oxygenation here of this vessel. This will change the gradient in magnetic field surrounding the vessel. So if we look at the situation now, cartoon-wise, we have the artery supplying oxygenated blood into the arterioles. Then we come to the capillary bed, and we have in the capillary bed, we have all our brain cells. Here we'll focus on the neurons. Then we have the venules, and finally the vein, that's the venous effluent. The whole thing that I've drawn here corresponds to a length scale of roughly 1 to 2 centimeters. So we have the feeding artery, the draining vein, and in between, at some point, we'll have the capillary bed. Now what happens if we apply an external stimulus? The neurons fire. As they fire, afterwards we get the blood flow response, and the blood flow response is much stronger than the oxygen consumption. That response results in an increased blood flow. In general, the increased blood flow decreases the deoxyhemoglobin content, and the venous blood becomes pink. That's actually, you can verify that. If you cut yourself in the foot, you're standing, then the blood that comes out is fairly dark. If you cut yourself in the foot is high up, and you put some warm pads around your leg, so you arterialize the blood flow, you increase the blood flow, then the blood that comes out of the wound is more pink. So what is this in time? So what this sequence of events, we have neurons firing, we have a change in blood flow, we have an increase in oxyhemoglobin in the veins, a decrease in deoxyhemoglobin. How does this look over time? So we're going to look here at the fMRI signal, T2 star weighted signal. We have the stimulus, which was applied here in the green bar, and a few seconds later we have a change in the T2 star weighted signal. Notice here the scale, it's percent change, so it's on the order of a few percent, the signal change. It's a big enough change to capture it with statistical means. It's not a big enough change to be able to look at an MRI and say what you're thinking. So MRI as a lie detector may work, people are trying it, but in general, you don't need to worry if you go into an MRI. One does not need to worry if one goes into an MRI, that one is able to read from the MRI right away what you're thinking. Okay, so we have now the bold response. We see a signal in T2 star weighted image, and the question is now, how is this used to image brain function? So here's a T2 star weighted image of the human brain, and if one now takes a task condition like looking at a light versus not looking at a light, takes the T2 star weighted images and takes the difference between these two time periods, then one gets an image like this. Fairly noisy. Outside here is no noise because it's been zeroed. And here we see an area that is fairly bright. This was in the early days how people looked at brain activation to convince the critics that indeed one can see brain activation, but this is no longer the way this is being done. But I'm showing it here for curiosity reasons. What is actually done nowadays is that one collects an entire series of images over time, over and over again, with special techniques that allow us to acquire this much faster. And so this is a series of T2 star weighted images, a time series. This is recorded. Here's, for example, the time series of a single pixel in a brain activation task, and you can see that the signal changes relatively periodically. These activations are linked to the task. In this particular case, there was 7 tasks applied, always the same task, and the change in T2 star weighted image here, the bold change, corresponds to the application of this task or the repetitive measure of this task. So now since what we are looking for is a change in pixel intensity, to say this is corresponding to a change in T2 star in underlying deoxyhemoglobin changes, what one does is one takes this time series, compares it to the measured time series, the task, and then does a statistical analysis. So here this means on and this means off. Actually it's state A and state B is the more correct term. So now comes the hard part, and that's a lot of statistical analysis. We're not going to go into the details here. One has to be very careful, very stringent in how it is being done, but what happens next? So what one gets then is a statistical significance image. That basically just says, I'm looking at the statistical probability that a particular pixel here is indeed following the activation pattern given by the task. And here we can see a higher statistical significance. This actually is not looked at directly. What is looked at typically is the thresholded statistical image. So after a certain z-score, statistical relevance score, one decides, yes, this pixel is indeed activated. It is responding to this particular task. And so one thresholds out the low statistical probability pixels and leaves only the high probability of statistical images. And of course, it's much more interesting to overlay this on an anatomical imaging to see exactly where the activation is. Otherwise, it appears just as a blob here. We won't exactly know where that is. So this is far more intuitive to see where one exactly is. So this is, in neuroscience, a very important imaging technique has become to study brain activation in systems neuroscience. It has, since the 90s, essentially replaced positron emission tomography as a means to look at brain activation. Now, we've talked about susceptibility difference between diamagnetic oxyhemoglobin and paramagnetic deoxyhemoglobin. But we have also other susceptibility effects to contend with, and this is particularly at the air-tissue interface, because tissue is diamagnetic, just like oxyhemoglobin, and the air, molecular oxygen, is paramagnetic. We have seen this. 20% air, 20% oxygen in the air produces sufficient paramagnetism in the air compared to our bodies, which are essentially water and therefore diamagnetic. So we'll have the same kind of transition between the paramagnetic species, which is the air around us, and the diamagnetic surrounding, well, that's our body, that creates changes in delta, in B0 at delta B0. And so here is a gradient echo image taken from the brain at a TE that's roughly T2 star. So, and now we can see here different black areas. This is a image of the brain. It's a cut like this, about here. And can you guess what these black areas correspond to? Well, this area up here is the sinus. We have here behind the nose, we have the sinuses, air-filled cavity, so it's filled with paramagnetic molecular oxygen. And here we have the ear canal that is also filled with paramagnetic oxygen. And this creates, on this type of image, creates a fallout of the signal because there's dephasing of the magnetization due to the delta B0. Here's another example of an implant. Implants, if their susceptibility doesn't match that of the tissue, they can produce the same effect. One example is, for example, tattoos. So how can we minimize these effects? And the signal here, we notice that the signal of the gradient echo is proportional to e to the minus te over t2 star. So we can minimize these effects by minimizing te. And if we do this, then such a t2 star weighted image will look like this. This is a gradient echo with a short te. However, we actually want to have T2 weighting to obtain contrast. We still wants to be able to see tissue contrast. So this solution is not applicable. And here comes the second solution that we will discuss now, and that is to use spin echo, which eliminates the effects of static field homogeneity. This is now coming up.